It's definitely on.
Four.
Yeah.
Okay. All right. Good evening. Hello. Hello to everybody. Thank you so much for coming on such a beautiful evening. And I think we've got to be quite impressed. We've got our second Regius Professor. I looked up how many there were. There are 70, so we've got a bit of a way to go. But Pastor's just been telling me that the post he holds was founded in 1540, and he is only the 27th person to hold the post since 1540. So after the Professor of Moral and Pastoral Theology, we have now the Regius Professor of Physics, reflecting the vocabulary of 1540. And we're very grateful for you taking time out of your holiday to do this for us. And you're talking about genomics. Sorry. I made a mistake already. That's my first mistake. We've got Morwenna, who's going to come and just talk to us a bit. There was an incident last month, and Morwenna is the president of the Anglesey Golf Club and also responsible for safeguarding at the club, and just wants to say a couple of words.
Hello. Hello, croeso I bawb, welcome. I'm Morwenna, I'm the president of Anglesey Golf Club. Coming to the end of my 2-year term, I've met a lot of you. Some of you have met me, some of you may have not met me. It's lovely to see the club holding these things. Fantastic speakers that you've had. It's really amazing how many speakers and what we've had. Sadly, last month I am the Safeguarding and Welfare Officer. And sadly, last month during Think, somebody thought it was okay to verbally abuse our young staff members. We do have CCTV camera, we have it. And I am asking a plea, basically. Please be kind to our staff members. You may be in a hurry to have a drink, you may be hot, but we have some fantastic staff here, and they do deserve the respect. So if you see or hear anybody else being disrespectful, please pull them up on it. We are accountable to Wales Golf. I am under the banner of Wales Golf as well. We do have a policy, a code of conduct policy, which is displayed in the cabinet there, or you can ask Nathan or any of them to see it. We don't tolerate abuse of any staff. We shouldn't. It's somebody's brother, mother, sisters, and whatever. We do not. We're a nice, kind, so please be kind. And one other little thing is, if you have dogs coming into the club, please keep them on a short lead under the table. We have some people that are scared of dogs, or there's been a couple of incidents where dogs have been hot this weather maybe and snapped. So please keep your dogs on a very short lead under the table. And that's it from me. Enjoy. Thank you very much. Thank you.
Thank you very much, Morwenna, and for welcoming us here each month. We really appreciate the facility, and it's a great place for us to do this, so thank you. And so, Patrick, over to you.
Well, thank you very much. So lovely to be here in Rosniger. Very honoured to be talking to you. We're actually, Owen is our landlord at the moment. And when he asked me to do this, I felt I couldn't really say no. So do we have any biologists in the audience? Excellent. Well, I think you'd be able to give this talk better than me. But I can't, I hope what I'll convey this evening is the revolution that's happening in our ability to understand DNA, particularly human DNA, and everything that goes on in your cells. This is the blueprint for your life, as you know. And I'll go over a few things that I think are interesting and exciting. when I was a medical student, the guy who taught me said if I learned one fact a day, then I'll pass the exam. So my hope is that you will learn one interesting thing today. So the picture's there. The first one's King's College, and you probably know Watson and Crick. They elucidated the structure of DNA, the secret of life, and they were both fellows at King's College. I don't work in the middle of Cambridge. I work at this place up the road. So in the 1950s, this was just farmland. I mean, it looked rather like the golf course, and it was less occupied than it was in Roman times. And now that's the biomedical campus on the edge of Cambridge. So it's got the headquarters of AstraZeneca. It's got the famous laboratory for molecular biology, and that really stemmed from Watson and Crick's work. And 12 Nobel Prizes have been won from that site, and it's one of the global centers of genomics. So DNA, that's what you inherit from your parents. It's a 4-letter code, GATC, and that gets turned into RNA. And the DNA, those 4 letters are grouped into 3. I mean, they're not physically spaced, but every 3 letters make something called a codon, and that codes for 1 amino acid. You've got 20 amino acids, and your genome contains about 20,000 genes, and they are about 20 to 20,000 letters long. So from tiny to absolutely enormous. So you have about 20,000 protein coding genes. And you'll have heard of genetics, which is basically understanding the function of genes one at a time. Your genome is all that DNA you inherit from your parents. So that's about 3 billion letters of code. And interestingly, about 2%, so a bit less actually, but about 50th of it is what codes for proteins. And the rest of it is partly just an accident of evolution. I mean, amazingly, it doesn't seem to cost us to have this extra DNA. Some of it is old viruses that have hijacked your genome, but much of it is the instructions for what gene to turn on at what time. So I think one of the extraordinary things is every single cell in your body can do everything that any bit of your body has ever done. So you could take any cell and it has all the instructions to make sperm, eggs, cartilage, a brain cell. So a real challenge is understanding all the instructions to turn genes on and off at the right moment. So I'm very lucky. I live about a mile from that site I showed you a picture of. So I just cycle to work every day. And on my way to work, I pass the houses of 2 of these guys. And they have developed and then refined the way that you can sequence DNA. And so after Watson and Crick worked out the structure of DNA in the 1950s, this guy, Fred Sanger, whose house I pass every day, he developed a way of sequencing DNA. And he did that in 1977, and he won the Nobel Prize for it about 3 years later. He's actually one of only 4 people ever to win 2 Nobel Prizes. So he's the only person to have won it twice for chemistry. He's a very modest man, refused a knighthood, a Quaker, and he has the Sanger Institute just down the road from us named after him. And the way that he developed this sequencing was, we have enzymes that copy the DNA. That's what needs to happen every time a cell divides. And he used one of those copying enzymes and then a radioactive base mixed in that stopped the chain. And then you could work out each time the chain stopped. And that was incredibly laborious. I also passed by this guy, Shankar Balasubramanian's house, and with his friend David Klenneman, down at the Panton Arms, a Cambridge pub, they had this idea that they could dramatically improve this process. So the first thing they did was, instead of reading one stretch of DNA at a time, they did millions in parallel, quite short reads. And I knew that was a really stupid idea which would never catch on, because how would you put all of the short bits together? Actually, with modern computers, that turns out to be quite easy. And then the other thing they did was to develop a way that you could wash the end of the chain off and let it go on a bit longer. So this absolutely revolutionized DNA sequencing. So Fred Sanger got his Nobel Prize for reading 5,000 odd bases of DNA, 5,000 letters of that GATC code. And then things moved along reasonably quickly. 7 years later, you could publish a paper in a leading journal with 170,000 bases. And then people had the idea of dividing up the DNA into bits and sequencing it in parallel. And you could get up to about 1.8 million. And then there was this extraordinary project to sequence the whole human genome. And that was really, I think, the first big biology project. So that involved particularly scientists from America and the UK. And to sequence the 3 billion bases of human DNA, that took $3 billion. And it took, I would say, 1,000 scientists working right across the world, probably for about 5 years to do it. Today, you can sequence a human genome, all 3 billion bases, in an afternoon for less than $500. And the extraordinary thing about that is, I mean, your DNA really does govern almost everything about you. Now, of course, that gets shaped by your experience, but actually, your experience is also shaped by your DNA. Your preferences are very strongly shaped by what you inherit. So now for less than $500, you can get the complete, instruction book for your life. It will govern how tall you are, whether you have blue eyes, brown eyes. It'll have a major impact on how intelligent you are. Not everything. Your education will be very important in that. So this is a graph of sequencing the human genome. And one of the things is that now we can use that reading system for DNA to read RNA. And you might remember that's the instructions that decoded from the DNA. So you can use a trick to turn that back into DNA, and then you can work out in every single cell what genes it's turning on and how strongly they're turned on. So I think this is leading us to a new way of thinking about medicine. And this guy, Leroy Hood, a very, very charismatic American, came up with this idea of 4 Ps medicine, which he was ahead of his time, but I think it is where we're heading. So medicine that is precise, personalized, predictive, and participatory. I'm a doctor, I still see patients, and certainly that's not what we do today. So, for many things, we will give you the same medicine, regardless of who you are, and we certainly won't look at your DNA. So it won't be personalized and it won't be precise. And on the whole now, we're still broadly reacting to illness when it comes up. And although I think doctors have got better at the participation, the extent to which we ask you what medicine you'd like, we could do better. So as we've got better at sequencing genomes, it has really led to a dramatic increase in the number of diseases that we recognize that are caused by single changes in a gene. Actually, that's not quite right. Usually these are caused by two changes, one in the gene that you get from your mum and one that you get from your dad. Because most genes, both copies are being read, and in a way that's a very clever aspect of evolution. So it means that you have a spare copy, if you like, that you can experiment with. Usually, when there's a change made, doesn't work so well, but the normal one will carry you through. But if you inherit a faulty copy from both your mum and dad, then you'll have a recessive gene disorder. So cystic fibrosis would be a common example in the UK of that. And over the last 20 years, I would say we've gone up from recognizing 100 or so diseases like that to 10,000. And of course, when you add that together, it's actually very common. So lots of people have rare diseases, and we're getting better and better at recognizing them. And you probably all know someone who's had a rare disease that doctors haven't picked up immediately, and it's taken some years to get to the right diagnosis. And DNA sequencing of whole genomes is changing that, and I'll tell you a couple of stories about that. And on the whole, these are lack of the right protein, on the whole. And increasingly, we have ways of putting a correct copy of the gene in, and then you can replace the missing protein. So that's gene therapy. And increasingly that's being done. So, for example, hereditary blindness. Now there are really good examples of gene therapy for some cause of that, or hereditary deafness, or haemophilia, which you've probably all heard of. And another important thing about this is, although there are many diseases that we still can't cure, if you do have a disease like this, then now it is well worth talking to a genetic counselor, and there are strategies for not passing on that to a child. So you can use something a bit like IVF. You can look at several embryos, and then you can pick ones that don't have the disease. So this is a short story from Cambridge. So these two pediatricians were interested. If we sequence the whole genome in children where something isn't quite right, can we work out what's going on? And to do that, they worked with the sequencing company to do it on a tiny drop of blood, so you didn't need to take a big blood sample, and so that you could get a result in a few hours, maybe a few days, from that 3 billion letters of code. And this has really been an extraordinary story. So if you take babies on the neonatal intensive care unit, where there isn't an explanation for them being poorly, then 1 in 4 of those turned out to have a genetic problem that you could identify like this. If you took the pediatric intensive care unit, so the difference there is the neonatal intensive care unit, if there's a problem immediately you're born, that's where you go. And then the pediatric intensive care unit is once you're a child, so any time after childbirth. So 1 in 3 children in the pediatric intensive care unit, this made a diagnosis. And in the neurology outpatient clinic, 1 in 2 children who didn't have a diagnosis were given one. So that's really extraordinary, and that's being rolled out now right across the country. And sometimes this is about not having to treat. So this is an interesting story of this baby, Oliver, and he had a very rapidly growing tumor on his leg. And previously, what would have happened is he'd have been given chemotherapy and surgery, and the chemotherapy would probably have meant that he wouldn't have been able to have children himself. Now, what the genetic analysis of this tumor showed was that actually it was a rare thing that grows for a period and then it stops and goes away. So they were able to just watch this and it got better by itself. So the precise treatments like the gene therapy I was telling you about, these are good because they are based on understanding the molecular cause. They'll often be curative, but they are, at the moment, unbelievably expensive. So the way this is working in the USA, typically, the cost of treating a single child, would anyone like to have a guess? Yeah, sold to the man at the back with the glasses. Peter, what's your guess? $1 million. $1 million. And there's some problems with that because, you know, I told you there are 10,000 different conditions. So, and of course, it also means that in less developed countries, it's impossible to think about it. So this needs a completely different approach to how we price these and how we roll them out and how we make them. Also, it's becoming clear that lots of common diseases can actually be subdivided once you know the genetics. And then you might be able to make a better treatment. So this is another story. This Professor Jean Abraham, with her colleagues, she set up a program where every woman who comes into our hospital with breast cancer, the cancer itself is sequenced and the blood. And cancer is a set of genetic changes. And once you know every gene that's gone wrong in the cancer, then you can make a much better approach to treating it. And so this program, which every woman pretty much signs up for, this has changed the management of women with breast cancer in about 40% of cases. And there is still huge scope for doing better than that. Sometimes it will show, for example, an inherited predisposition to cancer, like Angelina Jolie had, the BRCA gene. So a little bit about cancer. Mostly we diagnose cancer when it's very advanced. Often it's spread, and that's horrible to treat, and often we can't cure it. And a real vision is to identify cancer much earlier. And an extraordinary thing is that you can, once you can look at all the letters of code in every cell in your body, you can look back in a cancer, and this is like a barcode, all the change in the cancer, and you can ask, when did the first change happen? So this is a young adult in his 20s with blood cancer, and what it's showing you is that the original change that drove the cancer happened when this young man, was 6.2 weeks old. Now, what that means is that you have 20 years to identify those cells and do something about them. So the cancer that you get today, I mean, I don't know whether this is scary or reassuring, but we all have cells in our body that are most of the way to cancer. And I'm quite sure that over the next decade, we'll reach a point where we can pick those up. And one of the ways you can do that is every time a cell dies, then DNA leaks into the blood. And so you can pick up these small bits of DNA from the blood. And there's a lot of interest in, can you pick up cancers before they're lumps by doing that? This is another approach. So the swallowing tube, the esophagus, that's a nasty cancer that gets picked up late. And by looking carefully to see what the earliest changes are, Rebecca, who works in Cambridge, she has this lovely test where there's a little pill on a string. You swallow it. Someone in the audience may have had this if you have indigestion. And then you just pull the string back up, and it has turned into this little sponge. That takes some cells off the lining of the esophagus. Look at it under a microscope, and if it's expressing this protein, then you know that this is a pre-cancer, and you can just sort of freeze those cells off. Now, some of us are a little bit overweight, including me. There's 28% of grown-ups in the country are obese. 7% have type 2 diabetes, which is, I'm sure that someone in here has type 2 diabetes. And this has been revolutionized by Wegovy, Ozempic. But that emphasizes a whole spectrum of biology. So it turns out that the reason why we get overweight is we really can't control what we eat. And that's variable based on your genes. So if you're at the extremes, like these children shown here, where they're completely lacking something, then they have an unbelievable drive to eat. And those have been cured by specific interventions, this time of a small molecule, an injection or a pill. And I don't know, do any of you have Labradors? Yes, they're very hungry, aren't they? And actually it seems that they've been selected by humans for a couple of genes that give them the same desire to eat in an extraordinary way. And that was probably good for them where they were bred. So the first thing, it probably makes them very teachable, because they will do anything for food. And the second thing is that if you're by the water and you're a dog and you have to swim, being a bit overweight is clearly an advantage. And I also wanted to mention that if you think of risk of things like heart disease, I've talked quite a bit about these genes that have really big effects, but actually most of the genetic variation between us is small effects. And that's how evolution works. I mean, generally big changes will be selected against over time. But between all of us, there's this marvelous diversity. But all of that diversity contributes to different risk of diseases. And I think a really interesting thing is adding up, working out the small effect of lots of genes, hundreds to thousands of genes that contribute to your risk of heart disease or, say, dementia. And then you can, once you've sequenced your genome, you can add up all those little risks and get an incredibly accurate picture of your overall risk. Now, at the moment, I don't think we're reaching the point where that leads to me saying, the right treatment for Peter is different from the right treatment for Owen, but I think we will get there very quickly. I wanted to mention COVID quickly. So that's a trivial size genome, only 23,000 bases. I was involved with a consortium in Cambridge that sequenced the COVID-19 genome, not the original sequence. That was done in China, published very quickly, and that led to everyone else being able to sequence it. But at one point, we'd done half of the COVID sequences in the world. And you might say, if you've sequenced it once, why is it interesting to sequence it again? And looking back on COVID, I'm sure you'd rather forget it. I certainly would. But this was unbelievably powerful at the time, because these tiny differences in COVID between people meant that we could be absolutely certain where you had got your COVID from. So, for instance, in Cambridge, where there was quite a bit of trouble at one point because the townspeople were convinced that the students had brought the COVID in, and that the irresponsible students who were, not following the rules had infected the townspeople. But we were able to, I had no idea what the right answer would be, but it turned out that these were different trees of infection. And it also meant that we could pick up new variants very quickly, and actually the UK was very good on that, and that told you whether the vaccine was likely to be effective. And some other things I thought I'd mention very quickly. Blood groups, you all know about ABO blood groups. Actually, there are over 30 blood groups. And if you're someone who needs lots of blood transfusions, for example, if you have sickle cell disease or thalassemia, then you will get sensitized to all these groups that we regard as minor, because they aren't usually a problem. But if we've moved to doing comprehensive genetic testing on donated blood, and that means that we can match it much better, for example, to sickle cell patients. And that means that they don't get these reactions to blood transfusions. Just on sickle cell, I'm not going to talk about it any further tonight, but another amazing discovery of the last decade or so has been within those 3 billion letters of code, it is now possible to put in something that can search for a specific string of letters anywhere in the genome. And this will typically only be there once, and then it can cut that, and it can correct it. So increasingly, that means that we will be able to correct your DNA. And then I think a really amazing thing is clever people have now come up with ways that you can use redundancy in that genetic code to use new amino acids, new protein building blocks that have never been used by biology. So you can remove the redundant letters and then put in new bits of kit, and you can make cells that can make things that biology has never been able to make. And I think if you think of some problems we face, like, if there's an oil spill, could you make a bacterium that could just digest the oil? I wanted to also talk very briefly about your mitochondrial genome. So this is a tiny thing that is like the batteries in your cell. It's only got 16,000 bases. So I think that's about a 200,000th of the DNA you inherit from your mother and father. This only comes from your mother. It actually changes a little bit faster than the DNA in the nucleus. And the pictures I've put up here are related to my own mitochondrial DNA. So I may not look it, but my mitochondrial DNA is from an African slave in the 18th century. And her mother was taken over to Jamaica. And this handsome-looking chap was sent over as a 12-year-old, rather impecunious boy, was sent out with a parcel of ribbons to make his fortune in Jamaica. He did do very well, and he also married, he actually, unusually for a white man at the time, he married the daughter of this slave and brought her back to London. So some closing thoughts. I think it's very much a matter of personal preference, how much you want to know about where you come from and what diseases you might be predisposed to. I think with all this excitement about making precise interventions, we shouldn't forget simple things, like stopping smoking or taking more exercise, because those cost nothing, make you feel better. I mean, they may even save you money, and they will have more effect on your health than any of this. It is really important to cooperate globally, and actually the genome project, the Human Genome Project, was a brilliant example of that. I'm particularly excited by the possibility of now starting to understand how nature and nurture interact. You know, what can we advise our children to steer clear of, particularly for them? And I think an interesting challenge that this only goes a little way to addressing is how do we deal with the problem of falling to bits as we get older, which is happening to all of us. We're at different stages in the process, but this information can give us huge insights into that. So I'm going to stop there. Thank you very much for listening. This is Trinity College, founded also in the 1540s by Henry VIII when he created my chair. And if any of you would ever like to come there, then it would be lovely to have you for lunch. So, I mean, not to eat you, I mean as a guest. So that's it from me. Any questions? Of course, I'll do my best to answer.
Thank you very much indeed. Well, that offer of lunch became more attractive as you as you spelt it out. Can I just kick this off with one question? When you say correct the DNA, I mean that does raise, I imagine, quite difficult questions, perhaps for the Regents Professor of Moral Philosophy that we had on what a correction is and who decides what a correction is. I mean, there's quite a strong movement in the deaf people who have deafness, isn't there, that say that deafness is not a disadvantage and that they have a culture, a separate culture which they value and want to maintain and and in some cases don't want to be cured of deafness. So how do you decide what a correction is? Is that an issue you're aware of?
It very much so, and it's a fascinating area, I think. I think, with a consenting adult, we're in quite a good position. I mean, you can talk carefully through things. With children, I think it's much more difficult. I mean, parents would do anything for their children, but you cannot really know what the child would decide themselves. Then, of course, there are major societal questions. But I think if you have... It's one thing for people like me and you, Owen, who enjoy good health, to sort of pontificate about how we might feel if we had a disease that could be corrected. I think you can't really rely on that. I mean, if you want to think about how you might treat sickle cell disease, you really need to involve people with sickle cell disease. And one of the things that I have been deeply impressed by over time is patients I've looked after with rare genetic diseases who enter clinical studies where there's quite a risk to them. This is not changing their DNA. They are unbelievably altruistic. So even though they know that this study is quite unlikely to help them and might even make them worse, because it might make it better, especially in inherited conditions, because it might make it better for their children, they will try it. And I think another thing about this is, we have taken the genie out of the bottle and there's no putting it back. I mean, once you can do it, people are going to do it. A bit like, cloning a human in China, which was, I think in some ways more controversial. I think what it requires is discussion in highly intelligent groups like this.
Yeah. And just one other question from me. The way you describe it, Cambridge is very much at the forefront of this. I mean, I presume there are people in America who are also at the forefront of this. So how does that, I mean, is this genuinely an area where the UK is in the global lead, or is it, similar to America? How does it work?
Well, I think we are definitely at the forefront. China is moving unbelievably fast on this. So they contributed about 1% to the human genome, which they're incredibly proud of. But actually the best gene sequencing technology today is from the Beijing Genomics Institute. And they have, they can do it faster and at higher scale. And they've done some very, very interesting things, like made rice that you don't have to replant every year. And of course, the potential for this in agriculture, unbelievable.
Okay, thank you. Right, yeah.
Brilliant talk. I was really looking forward to it. Can you put it right up to your mouth? Sorry. Brilliant talk. I was really looking forward to it. A couple of things. At my age, I'm beyond help medically. So I'm depending on Elon Musk, because I think if he links my brain with his Gen 3 robots, which are actually walking better than me now, I'm in with a chance. But that's Neuralink and Gen 3. I'm serious about that, actually.
Yeah.
I haven't had a word with him yet, but the other thing is, I'm very concerned about glyphosate.
About?
Glyphosate.
Yes.
It's a worldwide deadly poison. It's more deadly than any of us know. Every one of us will have it in our garden shed and will have used it. It's incredibly deviously dangerous. It stops proteins folding properly. Yeah. It takes the place of glycine. Glycine is in all the proteins. Without the protein, we are protein. Without protein, we don't exist. All our cells are renewed continuously with new proteins. It will kill everything on Earth eventually. If you look at it, they say it doesn't kill humans, but it kills the bacteria in our stomachs, right? Which produce all the things that we need in our brain and everything else. I and everybody else depend on you guys looking at this and getting it banned.
Okay, glyphosate.
I'm sorry.
Yeah, and I do, that thing I said about the environment, I think is really important. So if you went back 70 or 80 years, people made these extraordinary discoveries about things that are really bad for you, like asbestos and cigarette smoke. And I completely agree with you. I think there will be things that we are exposing ourselves to that are much more dangerous than we realize. And one of the slight problems of the revolution in genomics and genetics has been that biologists have concentrated on the genetics because we can get absolutely precise facts really quite easily. And working out, whether exposure to glyphosate, 20 years ago is what caused your Parkinson's today, that's more challenging. But I do think we're entering a new period when we're going to get much better at doing that. And it's very important, I agree.
Question over there, yep.
Well, thank you, Patrick. A fascinating talk. Thank you for delivering that to us tonight. I suppose one thing that occurs to me is that evolution has worked for billions of years through genetic diversity. And yet, what you're suggesting in terms of correcting our genome would suggest that we're driving towards a genetic singularity, where you have a right set of genes that mean that you live the longest you can, and maybe we all die at 99. Is that a good thing?
So I don't. I mean, that's coming from a similar direction to the point Owen made, which I answered rather badly, which is, eugenics is a terrifying thought, and that we might engineer ourselves to sort of a single perfect thing. So I don't think we will do that. I mean, actually, genetic diversity is really important. If you think of something like COVID, genetic diversity there is crucial, particularly in your immune response. So I don't actually believe that for most of our diversity there is a better versus a worse thing. So as I said, most of the effects of genetic variance on things like diseases are tiny. And when we're talking about genetic correction, that's only for really serious errors. And I don't think those serious errors are likely ever to have been helpful, really. Cystic fibrosis, you probably know, is an interesting thing because the mutation that causes cystic fibrosis, if you have 2 copies, having 1 copy was probably pretty protective against cholera.
Yeah, really.
And so that's probably why it's survived. But now that cholera isn't a major problem in the UK, probably you could get rid of that. And as I said, I mean these things like sickle cell disease, cystic fibrosis, they are pretty nasty diseases, and I think if we could get rid of them, that would be sensible.
Hi, I'm thinking about artificial intelligence. How far away are we from AI being able to modify our genome and potentially doing so in a sinister fashion?
Yeah, what a great question. So I'd really recommend a book by a friend of mine called Adrian Wolfson. So he talks quite a bit about this revolution in biology. And what he says is you're now reaching a point where you can engineer biology in a way that biology has never thought of, and you have artificial intelligence that can do things where we don't even have to think. And that could be absolutely terrifying. So we need to think very carefully about how we contain that. And I am really worried about this thing that once it's happening, you can't stop it. I mean, I don't think humans have been very good at banning things because they're too dangerous. So yeah, I do think it's quite a challenge. I think it will also be able to do things that we haven't been able to conceive that will be good.
Yeah.
So, because I've got the microphone, I'll have a question. Patrick, that was fantastic. Thank you very much indeed. I have been a clinical scientist, and I thought that all of your discussion about the disease processes and everything like that, really, really interesting. I'm also really interested in ancient DNA and the use of genomic data to actually give a spotlight on where we've come from. And I don't know whether in your job you've had any input into that or have any feelings about that and any interesting insights.
Well, it's a fascinating area. So one thing that is interesting, which you might have read about, is it looks as if we intermixed with Neanderthal, previous close versions of us for much longer than people recognize, and that we do have quite a bit of Neanderthal DNA in us. Interestingly, some more than others. It's also, I think, becoming clear that you can... So I think it's fascinating in terms of history. I mean, you can get really reliable information about population movements, civilizations, things like when did our predecessors learn to use tools, and what was it that changed in the DNA that enabled them to do that? Also, I think it is going to be possible, if you found a really good bit of dinosaur stuff, I mean, you quite possibly could recreate a dinosaur. You only have to be able to put together the DNA sequence, and I think you could take that with descendants of dinosaurs, and you could make a pretty good guess. And with a bit of AI, no, I would personally not want to, and I think that's getting into, and I have a lot of faith in humanity. I mean, I think on the whole, we'll end up making sensible decisions about these things.
Yeah.
Well, what I'm interested in is because you've got Trinity there, reminded of Francis Bacon's hope, which in a way you're following the line of, let's live longer and the pursuit of medicine. And I'm also reminded of Wittgenstein at Trinity. And I suppose, death is the evolutionary selector, really. That's the thing I wanted to, and like you mentioned with cholera, and obviously malaria and sickle cell, and when an adaptive function survives in certain environments. So just wanted to bring that. My grandson's cystic fibrosis, I've got a granddaughter who's T21, known as Downs. So clinical decisions as to screening and those things. All I wanted to bring in, which is not necessarily in your remit, but the ghost of eugenics is kinds of death, effectively. Never mind what lives we should support with the pure AI being or whatever. How do we, further that conversation, really, using the ways that you're... I'm not saying it's a promissory note and it's all going to go well and the cure will happen and we'll get the cause. But if we look at how environments change over time, where there's political pollution problems, say, in certain things that pass through generations. So it's just a comment, really, of how do we have your line with the caveat of living forever...
So I think there is something really important there, and I think people should die. I think living forever is a really, really bad idea. But I do think so. First of all, will we as people make sensible choices about that? But what I would love is that instead of healthy life expectancy in the UK now is, I think, 62. But life expectancy, that's for a man, 62. But life expectancy is about 80. And so the last 18 years will often be in bad health. Maybe with dementia or... So what I'm interested in is not how you can extend life, but how you can avoid that falling apart towards the end of life. So there's still a problem in that, if you're completely healthy, will you be willing to die? And I think that's going to be difficult. But I do think we could all agree that living in a state of ill health towards the end of your life, it is something we really would like to avoid. And I do think we will be able to make real progress in that, partly by incepting things like cancer. Partly, I think it will be possible to protect brain cells as you age. And one of the most amazing things to me is young people, if you injure them, they bounce back almost immediately. Their ability to, heal, and that's one of the most striking things as you get old. Your ability to bounce back gets less and less. So what I would like is healthy aging, but then I would like people to be dead. Including me.
Thank you, Patrick. That was a really interesting talk. Thank you very much for that. What do you think about Iceland and they're trying to eradicate those with trisomy 21 to make a perfect society?
I'm afraid I haven't been involved in that at all, and I don't know. I think I would want, as I was saying in response to an earlier question, I think the right people to think about that are people with trisomy 21.
Yes, as my daughter here has trisomy 21 and is amazing.
Exactly.
And it's important to have a diverse society.
I agree.
An inclusive, diverse society. Because it'd be boring if we were all the same.
No, I certainly agree with you. I think, and some of these areas, I think, are going to raise really important questions for all of us.
Yeah. Okay. Thank you very much.
Thank you. Anyway, yes. Okay.
Good evening. I wonder how you think about people who are being kept alive when they want to be dead. And it's one of those things that I could tear my hair out for.
Well, I mean, personally, I'm quite supportive of sort of voluntary euthanasia. I mean, I think if people want to be dead, and if that's really carefully thought through, I would not want to stand in their way. And actually, a few years ago, he was someone I knew reasonably well, and he had motor neuron disease, and he felt in a complete in a position that he couldn't cope with and couldn't manage, and he wanted to go off to Dignitas in Switzerland, and I decided that I would go with him if that was the only way that he could sort of carry through what he wanted to do. Actually, to my huge relief, he then sort of changed his mind, and he decided to stay in the UK. But I do think it was the right thing to do to try and help him, and I think it did help him a bit at a time when he was facing a very difficult situation. So I agree with you, but I mean these are also very difficult issues.
Got two questions. First you, and then yeah.
Right. I agree with that. You ought to be able to choose to die when life's too miserable. I was interested in what you're saying about rare diseases. We're friends of a family who have Ehlers-Danlos syndrome, and they've had a huge difficulty over the years. You know, they tell people they've got it, the medical profession, and they need different treatment. They say, "Oh, you can't have it; it's rare."
Right.
And now more and more people are being diagnosed with it, whether it's because of your system, but it is becoming more accepted that it really exists and really does cause you problems.
So more rare diagnoses are being made, basically.
Absolutely, many more,
Yeah.
and sort of with accuracy.
Yeah, yeah.
Doctors are not very good if they don't really understand things.
Hello. As an ex-doctor, I agree with you. But what I wanted to ask you was about medical students. I remember as a medical student working on various wards, including looking after haemophiliacs, for instance. And when you look at doctors now and the divergence of skills, orthopedics, how do you see medical education and the training of doctors changing given the profound consequences in early diagnosis of disease as a geneticist? Because it may be, I don't know, that certain diseases will disappear completely. I mean, give example, let's say diabetes, that might plummet. How do you see medical education and manpower, or woman power if you like, changing over the next few years with these huge advances in genetics?
Because you are responsible at Cambridge for
the medical students, yeah. So we produce about 300 new doctors a year. So actually, the way we train our doctors really, I mean, they might not agree with me. They might think that what we're doing is teaching them lots of facts and then getting them to regurgitate them. But what we're really trying to do is to teach them to think. And I do think our courses are, so it's probably, I mean, it's at the scientifically most difficult end of medical courses in the UK. And No, I don't want everyone who we train to be a scientific researcher, but we are aiming that every student we have is sort of scientifically curious and is interested in not learning, how we think about diseases today, but how we're going to, be changing over their careers.
Yeah, but I mean, you've got departments. You have departments just concentrate on specific diseases, and these departments are going to disappear or not disappear.
I do hope so. Yeah, yes. No, I do hope so. And it is interesting.
It needs a change, not just in medical education, but in doctors who are working now in various areas.
So I think that's actually much more important than what you were just asking about, the medical students, which is. So sometimes I wish that I was in the armed forces because if you have more family practitioners than you need in the armed services, then you can just tell them all to go off and train to be orthopedic surgeons, and they'll do it. But if you're running the NHS, that isn't what happens. I mean, you have a real problem with people you retraining. Or taking a different career path after a few years. And I think it's a very perceptive comment, because I do think medicine's going to change in ways that we can't foresee at the moment. And then you're going to have people who are in the middle of their career, and how are they going to change what they do?
Yeah, thanks for a brilliant talk. Two things. One, you were saying about the way you're teaching. It does seem that very little critical thinking is taught. It's all done by rote throughout schools.
Definitely not in our.
Not in yours, no.
Course, yeah.
But you see it in lots and lots of other areas. My comment about the genome thing: is it possible to organise something to alleviate arthritis?
Yeah, I mean that's so arthritis. Do you mean osteoarthritis or rheumatoid arthritis?
Either or both.
Either or both.
Because
so osteoarthritis is where the things just wear out. So there's not inflammation; it's wear and tear. And I think there probably the main thing is, can you get cells as they get older to make new cartilage? So at the moment, our approach to it, which has been reasonably successful, is that you chop out the bone and the cartilage, throw it away, and put in a bit of metal. But that is a long way from perfect, as we all know. I think there the vision would be, can you do something to repair rather than replace? And I think that goes to the heart of understanding the instructions that could make cartilage. Rheumatoid arthritis, these inflammatory arthritis that are driven by your immune system attacking a protein of yours, there's been unbelievable progress. So there the treatments are, just so much better than when I was a student. When you used to see people with crippling rheumatoid arthritis, and now generally by giving antibodies that interfere in some of the inflammation, you can really change it dramatically.
Yeah.
It's an observation really here, Patrick, and in danger of lowering the tone after a lot of profound questions and a great talk. You mentioned that these brilliant scientists came up with some solutions in the Panton Arms in Cambridge, and I think Crick and Watson came up with a lot of their great work in the fantastic Eagle pub in Cambridge.
Yes.
So should we be all spending more time in Imorva?
Definitely. That is absolutely right. Creative thought.
Right. I just wanted that reassurance from the Regis Professor. Thank you very much, sir.
It's interesting, Imorva's happened twice in the last two think sessions, hasn't it? I don't know why. But so very good.
Okay, well, I think with that trivial contribution, after that very, very substantial talk, thank you very much. And Patrick, that was you have such a command of the subject, and it was an absolutely fascinating talk. So thank you very much indeed. Great, really good.
How long do you think are you